Seth-Smith from the University of Zurich, and I'd like to explain about a story that we've been involved in where we were looking at a plasmid outbreak among Enterobacter isolates across Switzerland. And this first came to our attention through a collaboration that we have with the Swiss National Reference Centre for Emerging Antimicrobial Resistance. And they became aware of an increase in VIM-1 carrying Enterobacter in 2022. And here we can see the epicurve. Across 2022, we could see an increase in cases, definitely in the summer of 2022. And this epicurve here is coloured by sequence type. So what we can see from the different colours is that there is definitely a multiclonal outbreak, and this is not, this is not just a single strain. When we look in more detail at the core genome MLST, we can actually see even finer resolution. So the most number of strains come from the sequence type 133, which are mostly within one cluster. And this is a core genome MLST scheme based on over 600 genes, an ad hoc scheme that we made. And we set the threshold of allele differences at 10 alleles. And that represents about 90 single nucleotide polymorphisms. And here we can see that there are different clusters even within sequence type 133. And within the second most numerous sequence type, we can also see that there are many different clusters. So we've got VIM-1 in each of these strains within Enterobacter, but why on earth, given that they're so different genomically? So we decided to look at the context of the VIM-1, and we did some long-read sequencing to find out the plasmid context. And we identified a plasmid of over 300 kilobases, which was found very largely in many of these isolates. And this is a phylogenetic tree of 35 of the 39 plasmids. And here we can see that many of them are very, very closely related. This is a very small mutation level that we have here. And we also looked at how much of the plasmid was present in each of the strains. And we found that there's a large collection here, a large cluster, which contain over 90-odd% of the whole length of the plasmid at a very high identity. And then these clusters up here contain a bit less of the plasmid, so they may have had more rearrangements, but they're also quite closely related. What's interesting, though, among this cluster is that you can see that it's present within different sequence types, even almost identical plasmids, inferring that it did transfer between isolates between these sequence types. And not only that, but we also sequenced some Citrobacter and E. coli from within the same time frame that were also VIM-1 positive, and they also carry more or less the same plasmid. So where do the variations come in this plasmid? Here we look at what's missing from the reference plasmid in the other strains, and we can see that there is some variability. There are some loci that can be lost from this plasmid, including some of this battery of antimicrobial resistance determinants. So many, many antimicrobial resistance determinants are carried on this plasmid, and some can be lost over time, for example, the MCR9 and the TET-A gene. In these cases, of course, the VIM-1 is always present because that was an inclusion criterion for our study. So in summary, we have shown that with genomic surveillance of this particular outbreak, we can find out that it's multiclonal. We can look at the genomic level, the chromosomal level. We can look at the plasmid and find how that transfers, and we can look at the antimicrobial resistance level too. This plasmid is self-mobile. It's huge, and it carries a battery of nasty genes, antimicrobial resistant, and this is really a potential threat. But what we can say is that we have not found any more cases since the end of October last year, and we consider this outbreak over, but we will continue our genomic surveillance. And I'd like to thank all the co-authors on this study, also the NARA network who were involved in sending the strains to the National Reference Centre in the first place, and particularly the co-authors Marielle Groitman, Laurent Poirel, and Adrien Egli.